Alright, so we're going to cover the reproductive system now, and so we're going to cover both males and females. And so, in general, the reproductive system is a means to be able to reproduce ourselves. And it's also, we're going to talk about how we mature as from sexually immature individuals to sexually mature individuals and what starts happening once you hit puberty, etc. And so we're going to talk about some special cells that are responsible for procreation. We're going to talk about the process of producing those cells, and we're going to talk about some similarities and a good bit of differences between the two. So, the first thing we do is talk about some similarities. And so, these are called homologs. And what homologs are they are structures that are different in both individuals, males and females, but they come from the same embryologic origin or from the same developmental structures. And so they'll serve similar functions, but they definitely are different. So the first one that we talk about is the ovaries, and so ovaries are homologous to the testes. These come from the same structures embryologically, but they are obviously different in males and females. And then for both, are going to produce gametes and also sex hormones. The clitoris and the glands, or the top of the penis, is going to be from the same embryologic structures. And these both contain erectile tissues and tissues that have a lot of nerve endings, and these are used for sexual reproduction. And then we'll talk about and then the next one is the labia majora, which is homologous to the scrotum. These are going to be protective and covering type structures. And the greater vestibular gland in the female and the bulbourethral gland, both of these are going to produce mucus. For the females, it's primarily to produce mucus for lubrication, and for males, the mucus is used to help line or protect the urethra, or line the urethra to protect the sperm as they're exiting, and so it kind of lubricates the internal structures there for the male. And so a good way to kind of remember what homologous structures are is to think about the hyena. And so once you hear this story, you'll never forget what a homologous structure is. So a female hyena and a male hyena externally look almost the same. The labia on the female hyena are enlarged, and it looks very similar to the male scrotum. It's even fused, and so it looks the same. And for the female's clitoris in the hyena is enlarged as well. It looks almost identical to the male's penis, and the same kind of thing like with the female. The male hyena has the urethra through the penis, but the female's clitoris also has the birth canal through it. So her reproductive tract is also the same as or through that structure. So what ends up happening is the female hyena, whenever she gives birth to the babies, is she gives birth through the clitoris, and so it's very much the same as like if a male had to give birth through his penis, and so it's quite dramatic, I believe. And hyenas, they don't have large litters. They like most other carnivores. They'll have small litters of like one baby because more than one baby might kill the female. And so she'll have small litters, small pups, and they birth through the clitoris instead of through the penis. And so if you ever do a Google search for like a female hyena, you'll get images like this because the female hyena does have the enlarged clitoris. It looks just like the male hyena. And so also you get kind of some not safe for work images. But here is an image that kind of shows the way that the birth canal works in the female hyena. So the baby is in the uterus, and then whenever she's birthed, she comes out the clitoris here. And so it's quite dramatic. Typically students remember this for a while. And so here's some text that just goes through and highlights some of those similarities between the different structures. And so you can see those here. In general, whenever we start talking about the two systems, we're going to talk about primary reproductive organs and accessory or secondary reproductive organs. And the primary reproductive organs are going to be the ones that produce the gametes and the sex hormones. And the secondary organs, or the accessory organs, are going to be the ones that are like ducts that carry the gonads, carry the gametes away from the gonads. Or they could help nourish the developing young for like a female. So breasts are considered like a secondary or accessory reproductive structure because they're necessary for mammals to reproduce, but they're not involved directly in the act of producing gametes. So once you hit puberty, which typically happens. It's a little bit different for males and females. Females go through puberty a little bit earlier, but at this point in time, the sex hormones start getting produced by the gonads and the gametes start maturing. And so this is whenever sexual reproduction is possible. And whenever puberty is initiated, there's some research just to try to understand why puberty is initiated, and there's no clear answer that anybody has really found. What is known is that the hypothalamus starts secreting gonadotropin-releasing hormone, and what this hormone does is it stimulates the pituitary gland to secrete follicle-stimulating hormone and luteinizing hormone, and these two hormones start producing gametes and start maturing sexual organs. Or these two hormones don't produce the gametes, but they stimulate the gonads to produce the gametes, and then it starts the process of sexual maturation. So the gametes are, for a female, they're eggs, and for a male, they're sperm. And so the scientific term for eggs, we're going to use the term oocytes, because that is a more correct term. And for males, we'll still just call them sperm. So the females produce and mature, she produces several million eggs, and these are produced before birth, but we'll get to that later. But for the most part, she matures one per month. And so typically that's it. Whereas males will produce 100 million or so per day. We don't store them for very long. We keep them for a very short period of time, and then we will, eliminate them or resorb them and reuse those nutrients. Gametogenesis is the process used to describe production of gametes, and so we're going to talk about general gametogenesis first. And whenever we talk about this, we're going to be talking about meiosis, which you've already studied in Bio 1. So we don't go into it in too much detail, but after we talk about the general process, we're going to then, in the discussion of males and females, talk about specific gametogenesis, which is oogenesis or spermatogenesis. And so we'll go into those two whenever we get into specifics for the males and the females. So in order to understand this, what we have to start doing is understanding why do we have to undergo the process of gametogenesis. And so your DNA is stored inside of each of your cells, inside of the nucleus. And on your DNA are all of the genes for all of the different traits of your body. And so whenever you need to start trying to replicate this, what you will do is you will condense it down into things called chromosomes. And so all your DNA is contained on chromosomes. And you have 23 pairs of chromosomes in all of your body cells. And so 22 of those are called autosomes, which are the ones that contain most of the genetic information. And then the last pair are your sex chromosomes, and they also contain genes that control other characteristics. But this is going to have some region on it that contains something that helps determine whether you're genetic male or female. And so you will get one of these. The reason why you have 23 pairs is because you get one from each of your parents. And so you'll have two sets. And whenever you want to give this to your offspring to produce a viable child, you need to divide that up. So you can't give them two sets of your chromosomes, because then that would be cloning yourself. And then it would reduce genetic variation in the population, and it could be a big problem if there was like a disease or something that came through. And let's say that you made a clone of your mother, but your mother also had the disease, so that means you would then for sure have that disease. Also, if another disease came through and a particular gene gave you immunity or resistance to it, then that would be a beneficial thing to have. And so genetic variation is very important. So the terms that we are going to use to describe gametes versus regular cells are either diploid or haploid. And so all of the cells in your body that are not gametes are diploid cells. These contain 23 pairs of chromosomes. However, we have 23 pairs. We'll just use that as 2n to indicate that it's a pair. And this is going to be the cells that are your cells of your body, or somatic cells. And all of your gametes are going to be haploid cells. They just contain 23 individual chromosomes, not chromosome pairs. And so instead of calling it 2n, we'll refer to these as n. And so this is the process of gametogenesis is going to ensure that you give your child only one set of chromosomes. And the reason why that's important is because if you have too many chromosomes, your body doesn't develop. And so having too many chromosomes, for the most part, is lethal. It does happen in some organisms, and it's not lethal. In humans, certain chromosomes can have an extra set, and it's not lethal, but not the case for all of them. So some terms we're going to use to describe chromosomes are sister chromatids. And so I want you to imagine that you have one chromosome, and I'm going to draw out a picture here. Here is one of your chromosomes, and let's say you got this from your mom. And then you'll also get one from your dad. And these should be identical. Now, not completely identical, meaning that the genes can have some variation, but they should have the same types of genes on there. And so it's all organized in these chromosomes. So right here we're looking at a pair of chromosomes. You've got one chromosome and another chromosome from your other parent. So whenever you undergo the process of gametogenesis, your body is going to replicate these chromosomes. So you will end up having two kind of stuck together here at the centromere. And so what we've got now right here is sister chromatids. There is one pair of the same chromosomes. What we had before, let me go ahead and put it back. Right here we have homologous chromosomes. Homologous meaning they come from the same origin, but they aren't the same. You've got one from your mother and one from your father. And then a single one of these is just one chromosome. So the process of gametogenesis starts with meiosis. And meiosis is going to start with a diploid parent cell, and it's going to end up producing gametes, or haploid cells. And these haploid cells are only going to have one set of chromosomes. They're going to be 1N, not 2N. And so then these are going to be produced in the ovaries and in the testes, and then this is the whole process, and everything is stored within the ovaries and the testes. So the parent cells are in the ovaries and testes, as are the gametes. You won't have a cell from like your arm, for example, produce or start undergoing meiosis because that's not necessary in your arm. So what does happen in your arm is something called mitosis, and what mitosis is it's somatic cell division, which is one cell produces two identical cells. Now, you can like debate whether or not they're truly identical, because they're really not truly identical, because individual mutations can occur in the process of gene replication, but they are supposed to be identical. Under the ideal model, they are identical. Whereas in meiosis, the cells are all going to be completely different. You won't have two identical cells produced hypothetically ever, because there are some things that happen that ensure genetic variation in the chromosomes. And so meiosis is a type of sex cell division. And so instead of having one cell produce two cells, you'll have one cell produce four cells. The processes are similar, but instead of just replicating DNA and dividing once and having two identical cells, you replicate the DNA and divide once again, and then you'll divide those cells again to be able to get the single set of DNA, or the single set of chromosomes. And one important thing that does happen in meiosis is something called crossing over. And crossing over is going to be whenever you'll have chromosomes kind of, and it's good that my thing's screwed up there. We'll just use that as an example. The chromosomes are just going to switch places at one little part. And typically this happens in the telomeres, which is the part further away from the centromere. But it could happen anywhere, hypothetically. It could happen multiple places on a single chromosome. And it's just completely random. And so since it's random, statistically you can say that there are some areas where it's more likely to happen than others because of geographic location on the chromosomes. But for the most part, you can't really predict it. It just happens. And this is part of what ensures genetic variation. It makes sure that you don't have two identical cells ever produced for your gametes. So the first phase of meiosis is interphase. And so there's a good way to remember, or it's the way I remembered, to get the phases in order. I just remembered the phrase ip-mat. It doesn't mean anything, but just saying ip-mat helps me get those in order. So the first phase is interphase, and what happens in interphase is the DNA is going to be replicated in that parent cell. So the one parent cell, which had a single set of chromosomes, one from the mother and one from the father, and so what we'll do is there's an abbreviation that everybody typically uses, or a convention. That's just to use X as the term for one set of DNA. And so what ends up happening is it's going to replicate its DNA, and now you'll have extra copies. And each line of the X represents a chromosome. And so it's kind of like writing this out. And so you'll have one chromosome from the mother and the father, and this is going to replicate to form one copy of each one, or sister chromatids of each one. And so now you'll have cells with identical copies of DNA, and they're attached at the centromere, which is the middle of the X. So the first thing that happens is meiosis 1, or I guess the second thing that happens after interphase is meiosis 1. It begins after interphase, and so this DNA that was just copied needs to separate itself. And so it's going to result in 2 cells. And so here's a good overview. So in prophase 1, and in prophase 1 is where some of the biggest stuff happens in meiosis. So what happens is the homologous material is all going to condense, but it's in prophase 1 that whenever the chromosome synapse or come together, they are going to cross together. And so all of the homologous pairs are going to find each other. But you'll see here in the image that some of these have exchanged arms. And so you'll see that these red chromosomes have swapped arms with this blue chromosome. And so now you'll have genetic information that is unique, or a chromosome that is unique. It's never been seen before. And so this is a way that you can give, for example, like you can give your baby your mother's eyes and your father's hair. And that's because somewhere during crossing over, these 2 chromosomes have swapped places. And now that's a very simplified version of being able to state it. But they can just switch. And so this only happens in prophase 1 of meiosis 1. It doesn't happen any other time. And then in metaphase, everything lines up in the midplate. And then in anaphase 1, these homologous pairs are going to be pulled apart, or these sister chromatids, and so now you'll have sister chromatids here, or replicated chromosomes. And so you can see here that whenever telophase happens, the new nucleus forms around the genetic material, and then cytokinesis is the splitting of the cytoplasm. So these 2 cells here are going to enter into meiosis 2. And meiosis 2 is going to begin with these cells that have the 23 replicated chromosomes, and it's going to split them into individual chromosomes. So the goal is separating those sister chromatids. And then what ends up happening at the end is we'll have cells with only one set of genetic information. So here we'll have those cells that we just finished meiosis 1 with. And in prophase 2, the nuclear envelope dissolves, the chromosomes condense. Then in metaphase 2, the chromosome or sister chromatids line up in the middle here. And then in anaphase 2, the sister chromatids are separated apart. And so now you will have the nuclear envelope form around brand new individual nuclei that have only one set of chromosomes each. And you'll also look that these chromosomes were the result of crossing over, so you have brand new, never before seen footage of genetic information here. And so from that one parent cell we had before, we have now 4 brand new cells, all with unique genetic information. And these are going to be the cells that you will then use to help try to produce a new individual.